Hi, everybody. Welcome to our VacCAP webinar series. My name is Amaya Tucha, and I'm part of the VacCAP team, and I will be the moderator for today's webinar. Today, we're going to be discussing blueberry cell wall polysaccharide compositions of 3 distinct fruit firmness phenotypes with Dr. Marlee Trandel-Hayse. Dr. Trandel-Hayse is an assistant professor in post-harvest physiology of specialty crops in the Department of Horticulture at Auburn University. Her program focuses on the impact of pre-harvest systems on post-harvest quality, but analyzing shelf life, changes in internal flesh quality, volatiles, and other secondary metabolites in specialty crops. We are very, very excited to have Marlee today and very interested in her topic. And I'm just going to have a couple of housekeeping items before we let Marlee start with the webinar. First of all, if you can stay muted and with your videos off, we would appreciate that. If you have any questions, you can either put them in the chat or in the questions, and we will address all of them at the end of the webinar, so we can let the speaker give her presentation, and then we'll ask all those questions. We're going to be recording this webinar, and I will be posting on the chat where this recording is going to be posted, so if you want to review it or share it. And with that, I'll let Marlee take it over. Thank you very much for giving this webinar and for sharing this great information about this research that you've done.
Awesome. Well, good afternoon, everybody, and thank you for inviting me to give this webinar. My name is Dr. Marlee Trandel-Hayse, and I am a new assistant professor at Auburn University. But today we're going to be talking about some of the work I conducted as a postdoc at North Carolina State University on southern highbush blueberry. So we're going to delve into the plant cell wall, and we're going to talk about blueberry cell wall polysaccharide composition of 3 distinct fruit firmness phenotypes. I want to begin by going through an overview of the webinar. So we're going to start with an introduction and the gaps in cell wall research. I'm then going to move into some of the preliminary work that was conducted, and this preliminary work was helpful in choosing sufficient cultivars with phenotypic firmness variation. We're then going to move into materials and methods and start discussing some of the results. After we discuss some of the results, we'll move really deep into the plant cell wall. We're going to kind of go over cell wall 101. We'll delve into linkage assembly and polysaccharide classification. Then we'll end by piecing the puzzle together by my multivariate analyses and discuss the impact and future vision for work. So to introduce this topic, the research was funded by the VacCAP grant with specific focus on blueberry and cranberry. Now, the overarching goal of this grant was to discover DNA markers and fruit characteristics that maximize industry profitability and match consumer preferences of blueberry and cranberry. And of course, judging by the title, I focused primarily on blueberry. So for the remainder of the discussion, we will focus on southern highbush blueberry. Now, blueberry quality is critical for consumer acceptance, and our ripe blueberries vary widely in firmness. Unfortunately, the blueberry industry has been suffering from fresh market berries having inconsistent texture and sensory profiles. So what that means is blueberries being harvested from the same orchard at the same time, being packed into clamshells and being shipped to wholesale markets are varying in their firmness. So some clamshells are holding up well, while others are degrading rapidly. And research has been looking at why this variation is occurring. So some of the characteristics affecting quality include fruit morphology, turgor pressure, cell wall enzymes, chemical composition, fruit texture, and plant cell wall polysaccharides. So you ask, there's all of these different characteristics that could be affecting this firmness variation. Why did you focus on the plant cell walls? Well, pre and post-harvest measurements were only telling us part of the story. So my research also looked at blueberries during post-harvest storage, where I followed decay, leakage, and shrivel incidents. And these different characteristics were telling us a bit about the variation of our cultivars, but they were not telling us what was metabolically or physiologically occurring within the fruit and why texture was being lost. Now, blueberry texture was also analyzed, but unfortunately, it doesn't adequately predict post-harvest texture changes or quality loss. So that's really what pushed me into plant cell wall polysaccharides. Now, the cell wall is highly complex. It's composed of cellulose, hemicellulose, and pectin. And solubilization or degradation of these polysaccharides, along with changes of neutral sugars and loss of the middle lamella, have been specifically linked to fruit texture losses in berry crops. So that led me to hypothesize that plant cell wall polysaccharides and changes in their composition are related to phenotypic firmness variations. But before I really dove into the plant cell wall, I wanted to understand the literature and the gaps in blueberry cell wall work that has been conducted. So there's been a tremendous amount of work already performed on the cell walls, including total polysaccharide sequential extraction. Now, this extraction is a crude extraction that looks at total polysaccharides of pectin, hemicellulose, and cellulose. Now, these extractions have been assessed during ripening and storage of whole berry crops. And during storage, they indicated water-soluble pectic fraction increased and the hemicellulose and cellulose fraction decreased, indicating cell wall solubilization was occurring. So this further suggested that the cell wall was at play and solubilization may be responsible for some of the softening characteristics that we're seeing. Now, Blaker and Olmsted did some work on tissue microstructure analysis where they looked at roughly 10 cultivars, southern highbush cultivars varying in firmness and texture. So they looked at firm, crisp, and soft cultivars. And unfortunately, the microstructure analysis was inconclusive in describing what was going on in the internal quality. Now, Blaker also assessed cell wall material where she prepared clean cell wall material termed alcohol-insoluble residues, a blueberry peel and pulp with the same cultivars, and these AIRs failed to explain phenotypic firmness differences. And I speculated that she probably would have identified differences based off of these AIRs if she dove deeper into the sequential extractions. Now, other scientists have also looked at cell wall composition of 6 to 8 neutral sugars and whole berry samples, and they have followed cell wall immunolabeling of cellulose and of hemicellulose and pectin at harvest and through storage, and they did see increases in hemicellulose in berries stored for 28 days in cold storage. So again, that leads us to believe that the cell wall is at play. However, information is lacking on cell wall linkage residues, linkage assembly, and specific polysaccharide classification estimation. I believe that these missing links may be giving further incentives as to what is going on with the cell wall and which specific polysaccharides might be at play in firmness. So the objectives of my work, I wanted to quantitatively analyze cell wall sugar composition and linkage residue in blueberry peel and pulp, to further elucidate the influence of polysaccharide classes on textural phenotypes. My objective was to study the quantitative difference in cell wall polysaccharides of a crisp, firm, and soft cultivar at harvest. And I hypothesized that crisp and firm berries would have more hemicellulose and cellulose in the pulp, as these polysaccharides are less likely to depolymerize during ripening, leading to increased berry firmness. Now, before we get into the materials and methods, I want to talk about some preliminary data that we used. So I worked under Dr. Penny Perkins-Vesey and Dr. Massimo Iarrizzo for this particular project, and we collected 2020 preliminary texture analysis data on a large southern highbush population encompassing 68 cultivars. So we were able to look at that texture analysis. So we looked at pulp firmness of the 68 cultivars and skin strength of the 68 cultivars to really decide on cultivars that varied in phenotypic firmness. Now, if you look at these two panels, panel A and panel B, firmness differed in Indigocrisp. Emerald are firm and Jewel are soft cultivar, both in the inner pulp and in the skin strength. So this further suggested the decision of Indigocrisp as our crisp, Emerald as our firm or our industry standard, and Jewel as our soft cultivar for the remainder of the materials and methods. So now that I was able to pick my cultivars, I went on to blueberry harvest. Blueberry harvest was conducted at the North Carolina Blueberry Variety Trial in Castle Hayne. And I harvested from established cultivars of Indigocrisp, Emerald, and Jewel. This was a one-time harvest. Now, cultivars were replicated 4 times within the orchard, and there was 4 bushes per replication that were harvested from. Now, after harvest, blueberries were brought to the Plants for Human Health Institute, where they were rinsed with DI water and then split for either texture analysis or cell wall quantification. Now, I do want to mention that this texture analysis was not performed by myself. It was with my PhD collaborator, Hee-Duck Oh, and he worked very closely with Dr. Jiang Go, who was establishing these methods to perform the texture analysis on my blueberries. So we used a TA-XT2 plus texture analyzer, and we analyzed the epidermal, the hypodermal, the parenchyma, and the inner pulp firmness at various depths based off of Jiango's methods. Now, the rest of the samples went on to the preparation of the peel and pulp, cleaned cell wall material, which is termed alcohol-insoluble residues. And these 2 pictures give you an idea on what those cell wall residues looked like. So this is the pulp cell wall material, and this is the peel cell wall material. Now, after I prepared those AIRs, I went through a robust series of cell wall compositional assays. So I was able to split my AIRs of the peel and pulp into a randomized complete block design. Now, before I get into the depth of these methods, I want to comment that these cell wall methods are extremely laborious and time consuming. So from start to finish, these methods took roughly 12 days. So the method started with a series of carboxyl reductions. These reductions were critical to reduce the polysaccharide structure and remove small molecules, including proteins. From there, the samples were split 2 ways. They either went to semen hydrolysis or methylation and TFA hydrolysis. Now, semen hydrolysis was critical for the quantitation of 8 neutral sugars, 2 uronic acids, and degree of methylation in my samples, while methylation and TFA hydrolysis was critical for cell wall linkage assembly and polysaccharide classification. So they went through these robust series of hydrolysis and methylation. Then all samples were brought back together and they were further reduced with sodium borodeuteride. Now, this reducing agent is critical because it tagged the anomeric carbon or carbon number 1 and then reduced the sugars from ring to open form. Now, once this reduction occurred, I acetylated my samples for volatility and I injected them into the GCMS. So after performing all of these methods, we were able to sift through some of the results. And I want to start by talking about the texture parameters and how they differed among the cultivars. So we did analyze 4 texture parameters: the epidermis, the hypoderm, the parenchyma, and the inner pulp. And the epidermis and hypoderm are more related to peel texture parameters, while the parenchyma and the inner pulp are more related to the pulp texture parameters. Now we can see in the epidermal firmness that Indigocrisp and Emerald had the highest overall firmness in the epidermal layer, while Indigocrisp had the highest firmness in the hypoderm, the parenchyma, and the inner pulp. So what this suggests is that Indigocrisp does have a distinctive mechanical texture compared to the other cultivars. So we were seeing all these significant differences in the texture parameters, and I wanted to know why. The story was still not lining up. And so that's why I dove deeper into the plant cell wall. So my emphasis was on the cell wall. My methods were comprehensive and can estimate specific classes of pectin and hemicellulose. So all of these polysaccharides and their interactions were followed through these comprehensive methods. Now, these polysaccharides aid in cell wall adherence, plant defense, and internal flesh quality. I want to talk just a little bit about these polysaccharides. So pectin is known to be the glue between the cellulose and the hemicellulose matrix. Subsequently, pectin is the first to be solubilized or depolymerized due to cell wall enzymes rapidly targeting the backbone of pectin. So we do tend to see pectin being lost first. Now, hemicellulose is thought to be the most responsible for internal flesh quality, while cellulose is the most stable and least likely to be degraded. So we have this general understanding of the cell wall, and I want to take you all a little bit deeper into the various components of the cell wall. So we're going to spend a moment talking about pectin. Here we see a total pectic polysaccharide. Now, within pectin, there are 5 classes of polysaccharides. We have homogalacturonin, rhamnogalacturonin 1 and 2, xylogalacturonin, and apiogalacturonin. Now, homogalacturonin is known to be the most abundant pectic polysaccharide, encompassing roughly 60% of the pectins in the cell wall, while xylo and apiogalacturonin are typically at less than 10%. Now, to make these cell walls even more complex, pectin is the only one composed of a galacturonic acid backbone that runs through the entirety of the pectic molecules, while glucuronic acid is a uronic acid predominantly found in rhamnogalacturonins, which are polysaccharides that are more complex and branched. Now, these pectic polysaccharides are composed of 17 neutral sugar building blocks. 12 of those are identifiable on the GCMS, with 8 of these neutral sugars and 2 uronic acids known for their specific importance in the cell wall. So hemicellulose has a similar story. There are 6 total classes of hemicellulosic polysaccharides. We have arabinan, type 1 and 2 arabinogalactans, heteroxylan, heteromannan, and xyloglucan. Now, arabinans, arabinogalactans, and xyloglucans are the hemicelluloses that are known to connect directly to the pectic network. And typically, the neutral sugar side chains on these hemicelluloses are long, highly branched, and composed predominantly of glucose, galactose, and arabinose. Subsequently, glucuronic acid is a uronic acid that is predominantly found in pectin, but also does play a role in building heteroxylan. So we can see how difficult and complex it is to discern where these neutral sugars and uronic acids are coming from and what polysaccharides they're building up. And I do want to make a comment here again that hemicelluloses are thought to play the largest role in firmness. So that moves me into stating the cell wall is extremely complex. All components of the cell wall are intricately connected by chemical bonds of neutral sugars and uronic acids. How they are linked or bonded together and the specific amount of classes of polysaccharides can impact blueberry texture. Now, in preparing for this webinar, I dove into a dissertation from UC Davis, and I stumbled upon this really beautiful image further elucidating the complexity of the cell wall and all of the interactions that are occurring. So we're going to move into talking about some of the differences in the neutral sugars and uronic acids that I found. So differences were found. If you remember, I analyzed 8 neutral sugars and 2 uronic acids. I found significant differences in 1 uronic acid and 3 neutral sugars. Now, glucuronic acid was highest in the peel of Indigocrisp. Glucuronic acid is known to build pectic polysaccharides. However, the high abundance of xylose and glucuronic acid in Indigocrisp peel suggests that there are higher amounts of heteroxylan, a hemicellulose. Now, arabinose and mannose were 2 neutral sugars that were highest in the peel of Jewel, our soft cultivar. Now, arabinose builds arabinans and type 1 and 2 arabinogalactans, while mannose builds heteromannans. And both of these neutral sugars also play a role in pectic polysaccharides of rhamnogalacturonin. And finally, glucose was highest in the pulp of emerald, our firm cultivar. And glucose is found in many polysaccharides, but it does predominantly build xyloglucans. So we were able to gain inferences about this neutral sugar composition and the potential of what polysaccharides they might be building. However, linkage assembly was needed to confirm the orientation of these neutral sugars and specifically how they build the polysaccharides. And that is why methylation and TFA hydrolysis was a critical step needed for linkage assembly. So I want to speak a little bit about the importance of linkage assembly. Linkage assembly allows me to identify linkage residues, or the single sugar units with the bonds that link together to form these complex polysaccharide structures. Essentially, linkage analysis allowed for the identification of polysaccharide classes. So I want to speak a little bit about how linkage assembly works. Here we have that total pectic polysaccharide. We have that galacturonic acid backbone. And if you look over to the right of the screen, you see how that galacturonic acid backbone links together. So here we have a galacturonic acid that links from carbon 1 to carbon 4. Now, through methylation and TFI hydrolysis, we add methyl groups to all of the free carbons that are not already bound to other parts of the cell wall. So methyl groups are added to the free carbons. We do a gentle hydrolysis that cleaves the bonded galacturonic acid together. And we're able to identify that galacturonic acid was linked from carbon 1 to carbon 4 through that gentle hydrolysis. So with linkage analysis and linkage assembly, I identified 56 linkage residues from my sugar standards, and 45 of those were identified in my blueberry peel and pulp tissue. So speaking about the linkages in highest composition in blueberry, there was several linkages in high composition that primarily encompass glucose, xylose, and arabinose. Now, 6-gluc-P, a glucose linkage, is typically found in oats and wheat and has never been identified before in blueberry. Now, 4,6-gluc-P and 4-gluc-P have been previously identified in grape and were found to build hemicellulosic xyloglucan. And 2,3,4-xyl-P and 3,4-xyl-P are important residues that build heteroxylan and xyloglucan. Now, the high abundance of the linkages noted above provide further evidence that hemicelluloses are an abundant polysaccharide in blueberry fruit. But I wanted to go deeper. I wanted to determine the abundance of these polysaccharide classifications, and I was able to estimate those from the 45 cell wall linkage residues. However, these cell wall calculations were no easy feat. And I was able to use a protocol from Nature Protocol by Pedalino et al. And I will say that she was the holy grail in calculations of these polysaccharide classifications and just general cell wall methods that were optimized for blueberry. And Pedalino stated the expected linkage residues and monosaccharide assignment of the linkages known to build these polysaccharides. So I just want to give you a brief example of the complexity of these calculations, again, that were determined based off of Pedalino 2012. So Pedalino was able to distinguish the specific polysaccharides like arabinan or arabinogalactan type 1. She was able to determine the expected linkage and the exact monosaccharide assignment of those linkages from those polysaccharides. So we have 5-ara-F, 2,5-ara-F, and 3,5-ara-F that are all assigned to arabinan. Now, some linkages are actually assigned to other polysaccharides, and she was able to determine the branch point equivalent or the addition and subtraction of these linkages equating to one specific polysaccharide. So I was able to follow her monosaccharide assignment and calculate the polysaccharide classification and the peel and pulp of all of my blueberry tissue. This was very challenging, very time consuming, but I was able to estimate the 6 classes of hemicellulose, the 2 classes of pectin and cellulose. So here we see the estimated calculations based off of Pedalino 2012. We have my crisp cultivar, my firm cultivar, and my soft cultivar. And there are a couple of highlighted numbers here in this table, and I just want to speak on those for a second. So in Indigocrisp pulp, we had arabinan, heteromannan, and xyloglucan that were highest in the pulp, and these are hemicelluloses that are known to aid the integrity of the texture. Now, rhamnogalacturonin 1 was subsequently highest in the peel of the soft cultivar, and if you remember back to my discussion on pectin, pectin is most likely to degrade, and specifically rhamnogalacturonin is usually targeted first by enzymes and has been most related to softening in other crops. So I was able to estimate the polysaccharide classifications in my blueberry peel and pulp, but I still had unanswered questions. So I wanted to delve into this data a bit deeper, and I wanted to know which texture parameters were most important in distinguishing the phenotypes, which sugars and linkage residues were most related to the peel and pulp texture, and what sugars or linkage residues should we be targeting in breeding programs. Now, I did this by multivariate analysis, and I first performed a principal component analysis to see if my cultivars and tissue types clearly differentiated or separated among all of the texture and cell wall parameters that were measured. And if we look at this principal component analysis, we do see a clear separation. First, we see a clear separation of tissue type. So tissue type, peel, and pulp clearly separated from one another. Then I wanted to look at the specific pulp parameters. And I saw that all three of my phenotypic cultivars separated clearly in the pulp, while in the peel, the crisp and firm phenotype clearly separated from the soft. So that gave me further incentive that there are specific texture parameters or cell wall residues that may be at play. And that's what pushed me to perform a hierarchical cluster analysis to identify the cell wall and texture parameters most related to the phenotypes. So we're going to first look at the pulp cluster analysis. And I do want to make a comment here that the purple indicates enhanced or increased abundance of the parameters, while white indicates suppressed or decreased abundance of those parameters. Now, in this hierarchical cluster analysis, we have two clusters of interest. We have cluster 1 and cluster 2. Cluster 1 indicates all texture parameters and some cell wall residues were enhanced in the pulp of the crisp cultivar, while cluster 2 indicates 4 cell wall parameters were enhanced in the pulp of Jewel. Now, the cell wall parameters enhanced in the pulp of our crisp cultivar are specifically known to build arabinan and xyloglucan, while the cell wall parameters enhanced in the pulp of our soft cultivar are specifically known to relate to pectin depolymerization, and they build arabinogalactan and rhamnogalacturanin. Remember, rhamnogalacturanin is a pectic polysaccharide that is most likely to degrade. So this is a very intensive analysis with a lot of information that can be taken from it. What does this analysis mean? Remember, all texture parameters were enhanced in the pulp of Indigocrisp. This indicates further that Indigocrisp does have a distinctive texture when analyzing with the needle probe. And if we remember back to the texture parameters, the hypodermal and the parenchyma were significantly highest in firmness in Indigocrisp, suggesting that these two parameters were most effective in discerning texture in blueberry crops. Now, the hierarchical cluster analysis was consistent with the polysaccharide classifications. Indigocrisp was highest in arabinan and xyloglucan, and arabinose, T-ara-F, and 2-xyl-P are specific sugars and linkage residues known to build these hemicelluloses. And these hemicelluloses are considered tightly bound hemicelluloses, so they are less likely to be degraded after pectin is solubilized. Now, the soft cultivar was abundant in 4,6-gal-P, 2,4,6-glu-P, and 2,3,4-ara-P, which build arabinogalactan and rhamnogalacturanin, a pectic polysaccharide. And the percentage of methylation is related to rhamnogalacturanin solubilization. So higher percent methylation typically indicates higher solubilization rates are occurring. Now, the key message from this pulp hierarchical cluster analysis is that hemicellulosic xyloglucan and arabinan are the most responsible for increased firmness in the crisp phenotype. Now, I gained a great deal of information from the pulp analysis, and I wanted to do the same for the peel. So we're going to be looking at a hierarchical cluster analysis of the peel. Again, remember, purple indicates enhanced or increased abundance of parameters, while white indicates suppressed or decreased abundance. And there are two clusters of interest in this analysis. Cluster 1 is related to enhanced components in Jewel peel, which is our soft cultivar, while some of these parameters were moderate in Emerald peel, our firm cultivar. And cluster 2 relates to enhanced cell wall parameters in Jewel peel. And something that was very interesting is that none of the texture parameters were related to any of these clusters. However, the firmness of the inner pulp was most related to the peel of the crisp cultivar. And we're going to talk a bit about this on the next slide. But I want to go back to cluster 1. These enhanced cell wall parameters in the peel of the soft cultivar are most likely related to xyloglucan and heteroxylan, and that percent methylation is showing back up, indicating pectin depolymerization was enhanced and is likely occurring in the peel of the soft cultivar. Now, again, looking at the peel of the soft cultivar, we have several cell wall parameters in cluster 2, and these cell wall parameters are related to arabinogalactan-rhamnogalacturanin interactions. So the communication between pectin and hemicellulose in the peel of the soft cultivar. So what does this peel cluster analysis mean? Now, the inner pulp composition was most related to the peel of the crisp cultivar. The peel firmness of the crisp cultivar, I hypothesized that the peel of the crisp cultivar is less likely to depolymerize due to the presence of hemicellulose in the pulp. So I speculate that pulp hemicellulosic content is protecting the peel from degradation. And in both cluster 1 and 2, we saw a high presence of pectin and hemicellulose interconnections in the peel of the soft cultivar. Xylose 2-xyl-P and 3,4-xyl-P specifically build xyloglucan and heteroxylan, while the increased abundance of the mannose linkages, the arabinose to rhamnose ratio, and the percent methylation indicate high presence of pectic side branches and arabinogalactan-rhamnogalacturanin interactions. So I was not expecting to see a high amount of hemicellulose in the peel, but it did make sense that we saw a higher presence of pectic side branches and rhamnogalacturanin interconnections due to the likelihood of rhamnogalacturanin solubilizing during ripening. So the message for this is that increased percent methylation and arabinose-rhamnose ratios in the soft cultivar indicate these cell wall interconnections are more likely to solubilize during ripening events, leading to whole berry softening. Now, I went through a great deal of information here, dove deep into the cell wall, and I want to talk about the impact of this work. So the linkages of T-ARA-F, 2-xyl-P, and 4,6-gluc-P are potentially the most important for us to be focusing on and upregulating in breeding programs for increased abundance of arabinan and xyloglucan, which are those tightly bound hemicelluloses, less likely to degrade. Now, high hemicellulose content in the pulp transition to high whole berry firmness, and these pulp polysaccharides seem to play a role in protecting the peel from degradation. Now, the type of hemicellulose can differentiate between firm and crisp cultivars. If you remember, the crisp cultivar did have higher amounts of arabinan and heteromannan compared to the firm cultivar that did have high amounts of xyloglucan. And this research indicated that it's not about one polysaccharide, it's more about how each polysaccharide interconnects and bonds with one another and how that depolymerization is occurring. For future direction, assessment of cell wall quantitative composition in a larger population is needed to further discriminate specific linkage residues and their importance in blueberry texture. Cell wall extractions are laborious, challenging, and time-consuming. We need new technologies to aid this analysis. Now, near spectroscopy is a promising alternative, but currently lacks a proper database. And I am very excited and curious to see the development of database for near spectroscopy. Immunohistology should be conducted in future studies along cell wall quantitative analysis to better understand how the cell wall interconnects and the relationship of cuticular wax to the peel cell wall composition may further elucidate water loss and softening variability during storage. I do want to go ahead and acknowledge the major advisors, Dr. Penny Perkins-Veazey and Massimo Iarrizzo, along with the cell wall expert, Suzanne Johanningsmeyer. I spent hours and hours and hours discussing this work and optimizing extraction methods. Without Dr. Johanningsmeyer, I would not have been able to fulfill this quantitative analysis. Big thanks to Hireko, a PhD candidate in the lab at the Plants for Human Health Institute, and the Blueberry crew at Castle Hayne, just for all of their help and their maintenance of this project. My long list of literature cited, and at this point, I can go ahead and open it up for questions.
Oh, Marlene, that was a lot of information and really interesting. Thank you so much for your presentation. I see we have one question in the chat, but please feel free, people, to just type your questions there. I'm going to go ahead and read it. So Desmond asked, for these three cultivars differing in firmness, was there any difference in the actual peel?
That is a great question.
Sorry, the peel thickness.
Yep. That's a great question. And so I had actually planned to do immunohistology and microscopy on these samples, but I was not able to do so because I ended up getting pregnant towards the end of my postdoc. So I cannot speak on peel thickness, but I do speculate that thickness did differ and may actually be thicker in the crisp cultivar.
I actually have a question. At the end of your summary, you said that more of this work is needed in a larger number of genotypes. But you said that it's incredibly time consuming to do this kind of work. So if you were to do this in a larger scale, what would you focus on, given what you learned out of this research?
Yes, I've spent some time thinking about this. I think if I was to do this in a larger scale, I would have to really think about replication and whether or not it's important to be looking at both the peel and pulp or the whole berry. I do not think that it is doable for us to focus on more than 5 to 6 cultivars with the current methodology, just due to the labor and time consumption. But if I was to focus on a larger cultivar set, I would probably focus primarily on the methylation and linkage assembly, because the neutral sugar analysis did give us inferences as to what polysaccharides were at play. They were not able to elucidate the exact structure, though. So I think in future work, we need to be focusing on linkage assembly and polysaccharide classification in the larger population.
Thank you. I have another question in the chat from Erin. Was there also a difference in the quantity of cellulose present on a fresh basis?
On a fresh base. So when I extracted the cell walls, I did not identify the weight of cellulose. I just identified the weight of the alcohol insoluble residues, and I did see significant differences in the weight of the alcohol insoluble residues between tissue type. Pulp had higher amount of alcohol insoluble residue than the peel, but due to the extraction methods of the cell wall, once I started the series of extraction, I was not able to pull parts of the AIR away from the extraction because that would tamper with my quantitative analysis.
Okay, here we have a couple of other questions. So from Pedro, how important do you think is the fruit calcium and boron content in the solubilization of the hemicellulose?
Yes, I actually, there is a professor from University of Florida that came and gave a seminar here just last week, and we spoke a bit about this. So calcium and boron are important dimers that play a role specifically in pectin communication in the cell wall. So I do believe that if we saw increases of calcium or boron dimerization with pectic polysaccharides, it would be lesser likely to solubilize both pectin and hemicellulose. Research on apple did see increased composition of calcium when apples were taken through storage, and that increased calcium concentration was related to increased fruit firmness. So I haven't been able to specifically look at calcium and boron in the cell wall or immunolabeling, but I do speculate that it plays a large role in protection of solubilization in pectin and hemicellulose.
Great. Janine asked, are any of the cell wall components you extracted and studied specific to blueberry, or would you expect to see these same components in blueberry relatives like cranberries? And do you expect the same components that conferred firmness in blueberry to be implicated in firmness in cranberries?
So very great questions. if you remember back to my discussion on the abundant linkage residues, the glucose linkage residues of 4,6-glucose and 4-glucose, those residues were previously identified in grape. So I do expect that some of these linkage residues would be in relatives like cranberry. And I do expect that some of these dominant linkage residues, like the glucose residues, the xylose residues, and the arabinose residues, would be similar in cranberry firmness. But we would have to further elucidate that by performing the linkage analysis and really understanding the role of those residues in other crops.
Great, thank you. I don't see any more questions. Oh, yep. Hudson asked, did you look at the bricks of soft and firm cultivars?
Yes, we did look at the bricks of the soft and the firm cultivars. It is in the manuscript that I just recently published. I know that the bricks was similar among all cultivars, indicating that they were ripe at harvest. We also looked at pH and titratable acidity as well to confirm ripeness.
Okay, well, I'm gonna wait a little bit to see if somebody else has another question. And in the meantime, I'm just going to post here where the recording of these webinars are going to be posted on our YouTube channel.
Thank you. Thanks to the USDA ARS group in Fort Pierce for coming and supporting me. I miss you all.
All right, well, it doesn't seem like we have any more questions. So we really want to thank you, Marlene, for a great webinar. I think we've all learned a lot about cell wall composition. This is great. As I said, the recordings are going to be posted if you want to go back. And I think that with that, we will conclude today's webinar.